So I apologize that I just had to start a new meeting. Unfortunately, my internet went out, which terminated my Zoom session, which terminated my ability to continue the lecture. But I'm back online, and so I'm going to continue the lecture where we left off here at the diencephalon. So the diencephalon, again, are these three paired gray matter structures, and all of which enclose the third ventricle. And so they're highlighted here in this sort of purple color, various purple colors. So first is the thalamus, and that would be this portion here is the thalamus. And then we have the hypothalamus, and hypo means below. And so as you can see, this region is the hypothalamus. It is below the thalamus. And then third, we have the epithalamus, which is located at the posterior portion, underneath and sort of behind the thalamus as well. So we're going to talk about each of these individually as we move through this lecture. So we're going to start then with the thalamus. Remember, that's this sort of bird eye looking structure here. The thalamus actually makes up about 80% of the diencephalon and contains a lot of different nuclei which project and receive fibers from the cerebral cortex. And so basically the main function of the thalamus is that it is a relay station for incoming information into the cortex. And so it's kind of like the gatekeeper. It basically sorts, edits, and relays all of this incoming information. For example, it sorts and edits and relays things like impulses from the hypothalamus, which would help to regulate emotion and visceral function. It also sorts through impulses coming from the cerebellum, which let me just get, so here's the cerebellum. It's going to sort through that information to help direct different motor cortexes. And then it also is going to sort through different information for memory or for sensory integration. So overall, it acts to mediate sensations, to also coordinate motor activities. It's also important for cortical arousal, learning, and memory. Moving on to the hypothalamus. The hypothalamus basically forms a cap over the brainstem and also parts of the inferior and lateral walls of the third ventricle. And it contains a lot of really important nuclei, like, for example, these mammillary bodies. So here is a mammillary body, which mammillary bodies are these paired anterior nuclei that act as olfactory relay stations. So relay stations for smells. Additionally, the hypothalamus has this structure called the infundibulum, which connects the hypothalamus to the pituitary gland. And at the very beginning of AP2, we will discuss the endocrine system, and the pituitary gland and the hypothalamus work together very efficiently in the endocrine system. So kind of talking about the major functions that are associated with the hypothalamus, it is the main visceral control and regulating center. So it is completely vital to homeostasis because, as I just mentioned, it has something to do then with the endocrine system. So in addition to that, it controls our autonomic nervous system. So things like controlling blood pressure and heart rate and the force of the heartbeat, also controlling digestive movement and the size of our pupils. These are all autonomic functions which are controlled by the hypothalamus. In addition, the hypothalamus initiates the physical response to emotions. And so this actually makes the hypothalamus part of the limbic system as well. And the limbic system is our emotional system. So pleasure, fear, rage, biological rhythms, and drives like sex drive. These are all responsibilities of the hypothalamus in terms of its coordination with other structures in the limbic system. The hypothalamus also regulates body temperature, regulates hunger and satiety, regulates water balance, regulates sleep-wake cycles, and controls the endocrine system functions by working together with the pituitary gland to coordinate the release of different hormones. So clinical connection here, why is this stuff relevant? The hypothalamus, if it is disturbed, it can cause a lot of different disorders. So for example, it can cause severe body wasting, which is basically just degeneration of tissues. So like muscle wasting, for example, is where the muscles begin to atrophy or get a lot smaller. Alternatively, it can also cause obesity, sleep disturbances, dehydration, and emotional imbalances because, as you can recall, based on its functions, any sort of disturbance in the hypothalamus could result in a disturbance of one of its main functions, which all would be associated with these different disturbances and diseases. And in terms of the hypothalamus, how can you actually damage it? It can be damaged by a lot of different ways, but some of the more common ways would be things like if you had a brain tumor pressing on the hypothalamus or infiltrating the hypothalamus. Also radiation therapy. Radiation therapy targets tissues and basically destroys them. And so radiation therapy can also be a cause of hypothalamic damage. Surgery, any kind of brain surgery, for example, the hypothalamus could be damaged. And then, of course, any kind of trauma, any kind of major head injury could result in damage to the hypothalamus, which could ultimately lead to many different effects. So finally is the epithalamus, and this is the most dorsal portion of the diencephalon. So whenever we were looking at the brain, we saw that the thalamus is here, and then here's the hypothalamus, and the epithalamus is towards the back, kind of closer to the cerebellum. And the epithalamus, the big point here is that it contains this thing called the pineal gland, which the pineal gland is part of the endocrine system because this pineal gland actually secretes a hormone called melatonin, and melatonin helps regulate our sleep-wake cycles. So as you guys have probably heard of, like, they sell over-the-counter melatonin to help with insomnia. It's sort of like an over-the-counter solution before having to go on any kind of medications like Lunesta or etc. But melatonin is a natural hormone. It's produced in the brain by this pineal gland. That basically the way this works. Just gonna like do a quick little lesson here. Is that you're being like, what are you drawing? Is that a sun? Yes, it is. So light blocks the pineal gland. Light keeps the pineal gland from releasing melatonin. However, at nighttime, when it's dark, that is going to lift the block off of the pineal gland, and then the pineal gland starts to release melatonin, which will make you go to sleep. This is also why you shouldn't watch TV or look at your phone before you go to sleep. So again, this is one of those tables that I told you are going to be present all throughout the PowerPoint. They're very helpful. I encourage you to study them for your exam. This is probably going to be one of the best resources that you can go to study for your exam. So now we've talked about the cerebral hemispheres, we talked about the diencephalon, and we're going to now talk about the brainstem before moving on to the cerebellum. So the brainstem itself consists of 3 major regions, and they're all, so if these are the hemispheres, and then the purple thing was the diencephalon, everything in green is considered brainstem. And so the most superior portion is called the midbrain, and then we have the pons, so midbrain, pons, and then the medulla oblongata. And structurally, it's very similar to the spinal cord, but if you remember, the distribution of the white matter and the gray matter is going to be different than what it looked like in the spinal cord, because there's those nuclei embedded in the brainstem. Basically, the brainstem is our sort of survival pass. It controls automatic behaviors that are required for survival. Things like breathing, things like heart rate, like sending impulses to the respiratory centers. All vital functions are located in the brainstem. It also contains fiber tracts, which are going to connect the higher brain regions to the lower neural centers, like in the spinal cord. And the nuclei that are in these cross sections of the brainstem are associated with 10 of the 12 pairs of cranial nerves. And we'll talk about the cranial nerves next week more when we get into peripheral nervous system. But for now, just know that these are nerves that are coming off of the brain. So, and all these little yellow structures that you see up here are the cranial nerves. So first, let's talk about the midbrain. The midbrain is located between the diencephalon, which is the purple structures here, and the pons, which would be like the second sort of structure here. So functionally, just by kind of putting these things into categories for you, this area, we'll say about there, would be midbrain, pons, approximately, and medulla, approximately, give or take from the side view. And so from this front view, the green structures midbrain, pons, medulla, okay, give or take approximately. So in terms of the midbrain, then it basically has several different nuclei, and these nuclei are things like the superior colliculi and inferior colliculi, which these are our visual reflex center and our auditory relay center. So this is important for seeing and for hearing. And then additionally, so this is a cross section of the midbrain. So whenever we are looking at the midbrain, if we just kind of cut it across this way, like you can see how it's cut here, this is going to be this top shear. We're looking at the structures that are in the midbrain. So in addition to having those superior and inferior colliculi, which are those relay centers, we also have what's called the substantia nigra. And this is functionally linked to the basal nuclei. Remember, we talked about the basal nuclei several slides sort of during the cerebral hemisphere sections. But the substantia nigra, there are these dark brown structures here. And these are clinically relevant because degeneration of this particular area is associated with Parkinson's disease. Additionally, the midbrain also has these, they're called red nucleus because they appear red, but these basically are a relay center for some flexion motor pathways, and they also make up part of the reticular formation, which we're going to talk about in a second. Okay, so moving down to the next section of the brainstem, which is the pons. So it would be located between the midbrain and the medulla. It's going to be composed of what are called conduction tracts. And so we have these longitudinal fibers, which are going to connect to the higher brain centers and also to the spinal cord. And then we're also going to have transversal or dorsal fibers, which basically relay information between the cortex and the cerebellum. So it's kind of the middleman between the cortex and the cerebellum. Additionally, one of the major things that you should know about the pons for the exam is that the origin of cranial nerve 5, 6, and 7 is located here. So here's 5 that you can particularly see in this image. You can only see, it looks like you can only see cranial nerve 5, but just know that throughout the pons, cranial nerves 5, 6, and 7 are going to arise. Okay, this is where they're stemming from. Some nuclei in the pons also play a role in the reticular formation, which we can see here in this area. And then some also help maintain the normal rhythm of breathing. So moving on to the final section, the medulla oblongata. The medulla is, oops, excuse me, sorry about that. It's a relay pathway that relays incoming information from the skin up to the brain. It also contains the cardiovascular center, which basically will adjust heart rate. And then also we have the vasomotor center, which is part of the cardiovascular center. So this is going to change blood vessel diameter. In addition, the medulla also contains the respiratory center, which has to do with respiratory rhythm and how fast and how deep you breathe. And the medulla also controls certain reflexes like coughing, vomiting, sneezing, hiccuping. And then here we will find nuclei for cranial nerves 8, 9, 10, and 11, or excuse me, 8, 9, 10, and 12. So again, here's one of those tables that summarizes the major functions of those different regions of the brainstem. And now we're going to talk about the cerebellum. The cerebellum is also called the little brain. That's kind of what cerebellum translates into from its Latin roots. It accounts for about 11% of the brain mass, and its job is to process input from the cortex and also from the brainstem, and also from sensory receptors to provide precise coordinated movement of skeletal muscle. So its major job is, I'm going to write it, maybe, balance. Cerebellum has the major big job of balance. The way that it helps to maintain balance is through these different activities. So it's going to receive some input from the cerebral cortex, and then it's going to initiate. So it receives these impulses from the cerebral cortex, which the cerebral cortex intends to initiate muscle contraction. It also is going to receive signals from receptors throughout the body. And then also it's going to process that, process signals from our vision and from our equilibrium pathways in our ear, which also ultimately, those pathways just continuously inform the cerebellum of the body's position and the momentum. And so this cerebellum then is going to calculate the best way to smoothly coordinate muscle contraction, and then it sends that blueprint of the coordinated movement up to the primary motor cortex and also in through brainstem nuclei. So it's a very complex but sophisticated pathway to motor movement. So that takes care of the 4 regions of the brain. And now we're going to talk about the functional brain systems. So we've kind of talked about the structural brain systems, but now we're going to talk more so about the structural systems. So the limbic system is a functional system, and it's going to include both cerebral and diencephalonic structures. So it's going to take some structures from the cerebrum and some from the diencephalon and form these sort of functional areas. And the limbic system, I've already mentioned it several times, but its job is to mediate emotional response and is also involved in processing memories. The reticular formation is a functional system whose job is to maintain alertness and also filter out repetitive stimuli and regulate skeletal and visceral muscle activity. So we're going to talk about each of these just barely in a little bit more detail. So again, the limbic system is a group of structures is kind of located in both the cerebrum and also in the diencephalon, and it includes some structures that encircle the upper part of the brainstem, and also has this region called the fornix. And so the fornix, you can see that would be, let me just draw it on here because sometimes it's hard to see where these arrows are pointing to. Okay, so the fornix, be that area. And the fornix is a fiber tract that links all of these different regions of the limbic system. Okay, and the limbic system, it processes all of this incoming information, and basically what it does is it relays that information to the hypothalamus, which is there. Because the limbic system and the hypothalamus are so closely tied together, remember back when I was talking about the hypothalamus, that the hypothalamus was very important for regulating the autonomic nervous system. So that's our fight or flight responses or our rest and digest responses. So disorders that have physical symptoms that are caused by emotional stress are called psychosomatic disorders, okay, because it's both an emotional disorder that has physical symptoms. And so we get the psycho part from the emotional portion of the disorder, and then the somatic part of the word comes from the physical symptoms that are associated with the disorder. So as I mentioned before, the limbic system plays a big time role in the emotional brain. And so, for example, the amygdaloid body is the structure that is going to recognize, so let me just draw a circle around it. That guy there. Okay, so that little structure there is the amygdaloid body. And this structure is responsible for recognizing angry or fearful expressions, and it's also responsible for assessing danger. And so this is actually the part that initiates or elicits the fear response. So it's super fascinating. And then we have the cingulate gyrus, which plays a role in expressing our emotions through gestures. And additionally, the cingulate gyrus is also responsible for resolving any kind of like mental conflicts. So cingulate gyrus would be this guy right here, okay, that I just highlighted. In addition, the limbic system also interacts with our prefrontal lobes, and so this allows us to react emotionally to things that we are aware are happening, and also makes us aware of emotional richness in our lives. So like understanding that emotions are a thing that can have an effect on you. All of this is thanks to our wonderful limbic system. So the second functional system then is the reticular formation, and the reticular formation basically just extends through the central core of the brainstem. So we can see it's sort of this pink-looking structure here. It has 3 bed columns that are going to run the length of the entire brainstem. So we have the wrapping nuclei, the medial group, and a lateral group of nuclei. And then it also has these axonal connections with the hypothalamus, which we see here, also with the thalamus, the cerebral cortex, with the cerebellum, and with the spinal cord. And so because it's so sort of widespread, the connections here allow the reticular formation to govern brain arousal. So that's kind of what I have here showing you guys, is that the sensory axons from maybe visual impulses, auditory impulses, and then like our touch receptors will actually, they're going to synapse on this reticular activating system in the brainstem. And then those neurons that are in this reticular system are going to send that sensory information to the cerebrum through the thalamus. And then we're going to have a continuation of that sensory stimuli get sent to the cerebrum, which is going to keep the cerebrum aroused and alert, right? So it keeps you thinking. It keeps you kind of, I don't know, not brain dead, I guess is the best way to explain it. So as I'm talking about this reticular activating system, this system is really important for filtering out repetitive or familiar or really weak stimuli. So believe it or not, 99% of all of the stimuli is not relayed to consciousness. Okay, so you're perceiving all of this stimuli, but only 1% of it actually gets sent to your brain to keep your brain considered in its conscious arousal state. So the reticular activating system then, because its job is to keep you awake and alert, would then be inhibited or sort of blocked by things like our sleep centers, right? So the whole purpose of going to sleep is so that our brains can rest. And so the sleep centers actually kind of deactivate temporarily the reticular activating system. Alcohol and drugs also would play an impact here. Any kind of depressants would block this reticular activating system. Severe injuries can result in a permanent unconsciousness, and the state of a permanent unconsciousness is called a coma. In addition to the function of keeping you alert and awake, the reticular formation also is important for helping control limb movements, and also has some vasomotor control, so control of your blood vessels, of the cardiac system, in terms of heart rate, rhythm, beat, so on and so forth, and then also with respiratory centers. So now that we've talked about some of these functional systems, we're going to talk briefly about some higher mental functions, and then I believe the final section is on disorders and diseases of the brain. So implementing language involves, I'm sorry, one second. So language, again, we kind of talked about this already when we talked about Broca and Wernicke's area, but I'm just going to touch on this again because it's so fascinating. The implementation of language, it involves the association cortex of a couple of different regions in the left hemisphere. So one of those regions then is called Broca's area, and Broca's area is involved in the production of speech. So, for example, if a patient had some sort of cancer or lesion in Broca's area, they would be able to understand words and understand language, but they wouldn't have the physical capability to speak. Wernicke's area, this region back here, Wernicke's area is involved in understanding spoken and written words. So unlike Broca's area, if someone has a tumor or some sort of lesion in Wernicke's area, they have the physical ability to speak, but the words that they are speaking would be completely nonsensical, that they wouldn't make any sense at all. There are similar areas to Broca's and Wernicke's area on the right side of the brain, but those areas on the right side of the brain, they're more so involved with nonverbal language components like body language and gestures and facial expressions. All of those would be then located on the right hemisphere, but in similar locations. So just to kind of put this in perspective, how this pathway works is, say someone's talking to you. That's kind of step one. Someone's talking to you and you are listening to their voice. Those sounds are sent through the auditory area up to Wernicke's area, where you make sense of all of those different sounds and recognize them as words, at which point information gets sent to Broca's area. Okay, so the information goes to Broca's area where you're thinking about what you're going to say back. And when you've decided what you're going to say back, information gets sent to the motor cortex, because remember, the motor cortex is involved in manipulating muscles for us to be able to speak or move our arms or whatever, in this case, speak. And so once you've decided what you want to say, that signal is sent to the motor cortex, at which point the motor cortex is going to stimulate the muscles in your mouth to move in order to produce the sounds that will create the words that you're speaking. So just the process of talking, of putting all of these steps together, as you can see, is a very sophisticated pathway that you don't really think that much work goes into speaking, but it certainly does. So next we're going to talk about memory. And memory, the definition of memory is that it is the storage and retrieval of information. And we have a few different types of memory. So for example, the declarative memory, this is memory of facts. So things like names, faces, words, dates, this is all related to declarative memory. Procedural memory refers to the memory of skills like playing the piano, for example, or knowing, I don't know, how to paint certain brush strokes, for example. Next is motor memory. And so motor memory refers to the memory of motor skills, like riding a bike or like, I don't know, writing the letters of the alphabet on a piece of paper. And then finally, the fourth type of memory is emotional memory. And this is the memory of experiences that are linked to a certain emotion. So, for example, whenever you see a rattlesnake in your garden or in your yard, your heart begins to pound. And so, the next time that you see a rattlesnake, maybe outside or maybe just a picture of a rattlesnake, your heart might begin to pound. And so, this is just an emotional memory, having a memory that's going to be tied to a certain feeling. So in terms of declarative memory, because this is one that I think we're all super interested in as students of people, you know, in education. So declarative memory, the memory of facts like names and faces and words and dates, or maybe the names of all the muscles or the names of all these different brain regions. This particular memory involves two different stages. And so we have the short-term memory stage, and this is also called the working memory. And short-term memory is really limited in its capacity. So on average, we can only remember 7 or 8 pieces of information in our short-term memory. Once you exceed that, some of the stuff gets booted. However, we have this other region of declarative memory called long-term memory. And this is where we hope all of our anatomy information goes into long-term memory. And long-term memory itself really has a completely limitless capacity. It's not limited by X amount of information. But in order to move things from short-term memory into long-term memory, you have to initiate this data transfer, if you will. It's like moving stuff from your computer onto a memory card. There has to be a data transfer. And so essentially we have different ways of transferring that data. Maybe it's something that we get really excited about, and so we just remember it better because it's something that we're really excited about and we really like to learn about or would really like to know. The other thing is rehearsal, repetitive nature of repeating something over and over and over again, like your address. It just becomes, you know, stored in your long-term memory. Additionally, one really useful way of storing things into memory is associating new data with data that you already have stored. And this idea of fitting these new facts into information that you already know is called memory consolidation. And ideally in this class, and in most of classes that are built upon a top of each other, all of the information that you're learning here is fitting into some part of your brain that remembers hearing about this or that in maybe the first week of the lecture, or the third week of the lecture, or something that you already know. And so a lot of times I try to phrase it and build these concepts based on things that you might already be familiar with, and hope that you're able to associate that memory of something that you already know with this new information, so that we can turn things in short-term memory into long-term memory. So some of the regions that are associated with memory include things like the hippocampus, some temporal cortical areas, the thalamus, and the prefrontal cortex. These are all areas that are involved with memory. So clinical connection here to memory. If you have damage to the hippocampus or to temporal lobe structures on either side, this could result in memory loss. If you have bilateral destruction, so if you have damage to both sides of the brain, particularly in the hippocampus or the temporal lobe that surrounds the hippocampus, this can cause widespread amnesia. You don't remember anything. There's a couple of different types of amnesia. So we have first the anterograde amnesia, and anterograde amnesia means that you don't lose your old information that you already knew, but you can't necessarily retain new information that's coming in. So basically, you might remember something that you studied like 5 years ago, but conversations that you had 5 minutes ago wouldn't be remembered. So you can't form new memories with anterograde amnesia. Retrograde amnesia, however, is where you don't remember anything from the distant past. So you can still form new memories, but you just can't remember old ones from far, usually distant past. Okay. So the next topic here is consciousness, and consciousness really just involves the perception of sensation, voluntary initiation and control of movement, and then certain capabilities that are associated with higher mental processing, like memory and logic and judgment. Consciousness is really clinically defined, basically on a continuum that grades behavior in response to stimuli. So alertness, drowsiness, stupor, and coma are the ways that we clinically define consciousness. But there's really still a lot of argument on consciousness. So some people believe that the idea of consciousness involves simultaneous activity of a lot of different regions of the cortex, whereas others believe that consciousness is superimposed on other neural activities. And then some other people just believe that consciousness, the idea of consciousness, is holistic and it's totally interconnected with every other brain function. So the idea of consciousness is really kind of obsolete in that we don't really know much about it, and it's very hard to measure. But in terms of the clinical connection to consciousness, except during sleep, any loss of consciousness signals that the brain is functionally impaired. So examples of this would be like fainting, which the technical medical term for fainting is called syncope. This is when there is a brief loss of consciousness. And most often this is due to inadequate blood flow to the brain, or it can be due to low blood pressure, or it can be all of these things would lead to inadequate cerebral blood flow. So low blood pressure, ischemia, which just means reduced blood flow, from hemorrhage, or also from a sudden severe emotional stress. And so sometimes, like whenever someone like receives really bad news, or something really dramatic happens, sometimes you hear of people fainting because they're just so overwhelmed with emotion. That would be an example of severe emotional stress. Alternatively, the term coma refers to an unconsciousness for an extended period of time. So this is not the same as deep sleep. Okay, they are completely two different things. In a coma, the oxygen consumption is lowered here. So finally, brain death refers to an irreversible coma. And something to kind of consider and think about is that there are a lot of ethical and legal issues that surround the decisions on whether a person is dead or alive. And so that's something that you might see in the healthcare field. There's just not, it's sort of a gray area, that there's just not a lot of legal basis here in medical support saying one way or the other. So that is just like something to consider, as well as you approach your time in the medical fields. So next, I'm going to just touch briefly here on sleep and sleep-wake cycles. So sleep refers to the state of partial unconsciousness, from which a person can be aroused by stimulating them, right? So basically this means that if you, shake somebody, they're going to wake up if they're just sleeping. So in this sleep state, the cortical activity is depressed, but the brainstem activity doesn't change. Remember, brainstem is required for basically our vital signs. So in sleep, that we actually have two major types of sleep, and these types of sleep are just defined by the patterns that you would see on an EEG. And so that's kind of what you see over here, is all of these different patterns. And so we actually have, again, two major types. So we have REM sleep, and we have NREM. And so REM means rapid eye movement sleep, and then NREM means non-rapid eye movement, and NREM is actually broken down into 4 different stages. So during REM sleep, the skeletal muscles, except for the eyes and the diaphragm, are completely actively inhibited. And so during REM, this is actually when most dreaming occurs. During NREM stage 1, so NREM stage 1, this is when relaxation begins, and we're going to see alpha waves. And so if a person is in stage 1, it's really easy to wake them up. So they basically just dozed off. In REM stage 2, we're going to start seeing these really irregular EEG patterns with what we call sleep spindles. And so these guys are both sleep spindles. Basically, these are just these really short, high amplitude bursts. And in stage 2, it's more difficult to wake somebody up. In REM stage 3, this is when you start to become a lot more in a deep sleep. And so this is when we're actually going to start seeing these theta and delta waves up here. And we're also going to see the vital signs start to decline. So things like respiratory rate are going to slow down, heart rate is going to slow down. blood pressure is going to drop a little bit. And so these are all things that we'll see in interim stage 3. And then finally in interim stage 4, the EEG is going to be dominated by these delta waves. And so all of these are delta waves. And arousal in REM stage 4 is going to be very, very difficult and will be associated then with things like bedwetting and night terrors and sleepwalking. All of these things are associated with stage 4 NREM sleep. So throughout the night, basically the pathway that we take through this sleep cycle is we're going to start here in the awake and then we're going to start to fall asleep. And actually the sort of the first the first part of the sleep, we go straight into stage 1 and we're here for a little bit. And then we are in stage 2 for a little bit. So anytime you see the flat line going across like this, this is going to represent sleep during whatever stage it falls in. Okay, so the first part of sleep, as I just mentioned, first part of sleep is this stage 1 period here. And then we drop into stage 2 for a little bit. And usually this is the first 30 to 45 minutes. And then we are going to shift into stage 3 and stage 4. These are what we call slow wave sleeps, because the frequency of the waves are going to decrease, but the amplitude, the height of the waves are going to increase. And remember, this is the period that heart rate and respiratory rate and blood pressure are going to change. Specifically, they're going to slow down. And then at approximately about 90 minutes into sleep, the fourth stage is going to come to an end, and all of a sudden we jump into REM sleep. And REM sleep involves temporary paralysis, except for the eyes and the diaphragm. And during REM sleep, kind of the opposite is going to happen when you compare it with stage 3 and 4. So in REM sleep, this is when our oxygen consumption is going to increase, heart rate's going to increase, breathing's going to increase. And sometimes in REM sleep, these rates are actually higher than what they would be when you're awake. And so most dreaming is actually occurring during REM sleep. Okay. As you can see, as time goes on, you can start to see that we don't get into those really deep stages of sleep, right? So say this is maybe between 4.5 and, I don't know, 7.5 hours in, we are not re-entering the stage 3 and 4. So we're not getting this deep, deep sleep. So why is sleep important? Slow wave, the slow wave sleep, so those stage 3, stage 4 really deep sleeps, they're presumed to be restorative. And kind of along the same line, people that don't have REM sleep tend to become moody and depressed because we believe that REM sleep might give the brain an opportunity to analyze the events of the days, and then work through sort of any emotional events or problems, but also eliminate unneeded synapses that were formed. So sometimes we think that the REM sleep allows us to dream so that we can forget unnecessary information that we formed during the day. It's also good to understand that the daily sleep requirements decline with age. So as you get older, stage 4 sleep will decline steadily, and you might not even get into stage 4 sleep after the age of 60. So relating this to why it might be relevant, we have some clinical scenarios to draw upon. So first is narcolepsy, and this is a sleep disorder where basically you just lapse into sleep from the awake state. So like 1 second you're awake and then all of a sudden you're sleeping. There's a couple different types of narcolepsy. So cataplexy is sort of a related condition, but this is a scenario where someone can lose muscle tone while they're awake. So essentially, it's where cataplexy would be where someone just like drops, hits the floor all of a sudden. We think that one of the reasons narcolepsy happens is due to these things called orexins, and basically these are just like wake up chemicals that are probably destroyed by the patient's immune system, leading us to believe this is an autoimmune condition. But basically by replacing orexin in the person, this is a potential treatment for narcolepsy. Alternatively, we have this other thing called insomnia. And insomnia just clinically is the chronic inability to fall asleep and stay asleep. And basically, this could be caused by depression, anxiety, maybe too much caffeine, computer or cell phone use that's too close to bedtime. And how you treat insomnia is actually by blocking this chemical called orexin. So we block orexin chemically with a medication, maybe an example would be like Lunesta. By blocking orexins, then you're able to fall asleep and stay asleep, thus resolving your insomnia. So in terms of, we've talked about all of the different structures here on the brain, but I think it's also important to understand that it's projected in a few different ways. So first is by the meninges, and these are the connective tissue coverings of the brain and the spinal cord. And so their whole goal is to cover and protect the central nervous system, and also contain the cerebrospinal fluid. And there's 3 layers of the meninges. So we have the dura, the arachnoid matter, and the pio matter. So starting from the outside, so we're going to say that this is the skin, this is where we're going to have our hair growing, okay, on the scalp. So here's the skin, and then we're going to have periosteum, and then we're going to have the skull bone, okay. Just below the skull bone, we're going to have the dura matter, okay. These are both dura mater. And then there's going to be a little bit of space in between the dura and the arachnoid, but next we're going to have the arachnoid matter. And the arachnoid, you can see from this picture that the arachnoid is this very sort of shiny, translucent layer. So this sort of shiny layer here that you can see through. And I can kind of put little dots all around the arachnoid. Okay, so this is all arachnoid. just beneath the arachnoid is called the subarachnoid space. Okay, and so for you guys to see, I'm going to just color it in. Okay, so all of this that I'm coloring in right now is subarachnoid space. And the subarachnoid space is what contains cerebrospinal fluid. And then finally, this last layer, the layer that really you can't really see it all that well because it's the one that has direct contact with the brain tissue, and that's called the pia mater. Okay, so the pia mater is lying directly on the brain tissue. Okay, and so I bring up the point here of meningitis. And so meningitis, now that you know what the meninges are, meningitis just refers to an infection within the meninges. Okay, some sort of infection within the meninges. And as you can imagine, any kind of infection, this could lead to the brain itself being affected by whatever is causing the meningitis, whether it be bacterial meningitis, viral meningitis, or so on. So I briefly talked about the subarachnoid space containing cerebrospinal fluid, but I just want to mention that cerebrospinal fluid is actually made in the ventricles, and most of it is going to be sent through this array of different sort of structures on its way. So ventricles are these fluid-filled chambers that are continuous with one another, and they're also continuous with that central canal in the spinal cord. And these are, again, they're filled with cerebrospinal fluid and lined by ependymal cells. That should not be a new word to you guys because that should be a review from the chapter on nervous tissue that we covered last week. So I'm highlighting that because it's something you should probably know. So the brain ventricles are going to then include 2 lateral ventricles. So here's a lateral ventricle. There's one. And then we're going to have a little area called an interventricular foramen that's going to, sorry about that, undo that. Going to be there. That's going to connect the lateral ventricle to what we call the third ventricle. So this shape here would be the third ventricle. And then we're going to have the next section called the fourth ventricle, but coming off of the third ventricle, we have what's called the cerebral aqueduct, and that's going to connect the third ventricle to the fourth ventricle. And then the fourth ventricle is going to lead down into the central canal of the spinal cord. So in terms of the way that cerebrospinal fluid circulates through the brain, it's actually made here in the choroid plexus in each lateral ventricle. And the cerebrospinal fluid is going to flow through this interventricular foramen. Okay. So we're going to flow through this foramen, at which point it is going to reach the third ventricle where the choroid plexus here can add more cerebrospinal fluid, at which point the cerebrospinal fluid is going to flow down the cerebral aqueduct until it reaches the fourth ventricle. So here we're in the fourth ventricle. And we have, as you can see here in the fourth ventricle, more choroid plexes that are going to add more CSF. And from here, the cerebrospinal fluid is then going to flow into both the subarachnoid space, which is what we're seeing here, subarachnoid space. It's also going to flow down the central canal of the spinal cord. And there are certain places in the brain called arachnoid villi, which is where, because we want to be constantly replacing the cerebrospinal fluid, essentially what happens here is that the cerebrospinal fluid that you just circulated can actually go into these little sinuses, which is in this dark blue structure here, and be reabsorbed so that new can actually flow through this area here, okay? So when talking about cerebrospinal fluid, just know that it's found around the brain and the spinal cord, and its purpose is to form a liquid cushion to provide buoyancy to structures in the central nervous system. It helps to decrease the weight of the brain because anytime you put something in liquid, it just makes it lighter, and also protects from blows to the head. So it kind of gives, instead of the brain just automatically going up against the skull bone, it gives it sort of like a liquid cushion. It also helps to nourish the brain, so there are nutrients that are present. And something, fun fact about cerebrospinal fluid is that it's actually replaced about every 8 hours, and is also cleaned by the choroid plexes, which are the ones that are making the CSF. Okay. So in terms of a clinical connection here, there's a condition called hydrocephalus, which basically this occurs when there's an obstruction that's going to block CSF circulation or drainage. And too much CSF is a bad thing because this is going to increase the pressure on the brain. In newborns, however, the skull bones are actually not fused yet. And so an increased pressure actually causes the head to enlarge. And that's what you see over here in this picture. In adults, though, because our skulls are fused at this point, this keeps the pressure inside, and this can potentially lead to brain damage because it might compress blood vessels and also crush the soft nervous tissue. And so the way that we treat this is just to drain this cerebrospinal fluid with a shunt to the abdominal cavity. So a shunt, it's basically like adding a pipe from the brain ventricles to the abdominal cavity. So all the extra fluid can just go into the abdominal cavity. So the third matter of protection here then is the blood-brain barrier. And this is actually very important because the brain really needs to maintain a constant internal environment. As we all know, the way that neurons work is that they have certain electrolytes inside and outside of the cells, and it's really important to maintain that balance. And so the blood-brain barrier provides a protective mechanism that helps to maintain a really stable environment. And the reason that it's able to do this is because we have tight junctions located in between the endothelial cells that are surrounding the brain capillaries, or the little brain blood vessels. And so in order to get to neurons, any kind of substances that are inside of the blood, they have to pass through 3 different layers in order to reach the neurons. So they would have to pass through the endothelial cells that we see here. They would also have to pass through a relatively thick basement membrane, which we see here. And they would have to pass through these parasites, which are the feet of the astrocytes here over on this side. Okay, so it's a very good thing to have this present, because our barrier is very selective. And so some of the things that the barrier allows to cross are things like glucose and things like amino acids and electrolytes, things that are relatively important to survival. However, it does not allow metabolic wastes, proteins, toxins, and most drugs to cross this barrier. And it's completely, unfortunately, the sort of downside of this all is that there are certain substances that it's just completely ineffective against. And so the blood-brain barrier is ineffective against fats, fatty acids, and fat-soluble molecules, oxygen and carbon dioxide, and also alcohol, nicotine, and anesthetics. Okay, so this is something that's good to know. All right, so the last section here are the brain injuries and disorders. And so first and foremost, we'll talk about concussions. A concussion is the least severe type of traumatic brain injury, because usually it just involves an alteration in brain function, but it's usually temporary. However, I say that cautiously, because repeated concussions can result in serious damage. And if you are interested in learning about this more, there is a documentary on Netflix that has to basically follows the story of a former NFL player. I believe it's Aaron Hernandez, and it's very fascinating. So if you're looking for something to watch on Netflix, I encourage you to watch that. It's really fascinating to see his brain scans, and also the transection of his brain. It's very, very interesting. So definitely check that out. So back to the traumatic brain injuries, we have contusions, which are basically just really serious concussions where the brain is actually bruised and neurological damage is present. Third, we have subdural or subarachnoid hemorrhages. So basically this is referring to a brain bleed. And over here in this picture, this is kind of what I'm trying to show you here. This image on the left, we have increase in density along this left cranial vault area. And what you're actually seeing is along this midline here, we can see a shift in this longitudinal fissure. And basically, what this means is that there is pressure building over here somewhere that's actually causing the brain to literally push up against this midline and thus change the move the midline over. And so that's actually what you can see over here on the right. This clearly shows all of this hypodensity area here. This lighter region, this is showing a subdural hematoma. So this is a blood that's in the space just below the dural matter, which would be, so subdural space would be between the dura and the arachnoid matter. There's some blood there, which is actually increasing the pressure because this particular, everything in white that you see here on the outside would be bone. And so blood can't necessarily go that way. So instead it just goes this way, and then it increases the pressure on the brain. All right, and that causes this midline to shift over. Finally, cerebral edema just refers to the brain itself swelling, which definitely can have an impact because there's nowhere for it to swell, and for the most part in an adult head. Next are cerebrovascular accidents, CVAs, and sometimes they're called strokes. Most strokes actually occur in the frontal lobe, and what happens in a stroke is that the brain tissue itself is actually going to die because the blood vessel is obstructed and not providing oxygen and nutrients to said region of the brain. That's the most common cause is due to a blood clot that actually ends up blocking one of the arteries that supplies the blood. And the middle cerebral artery is a very common one to be blocked. And so that's kind of what we're seeing over here. So this is the left side, this is the right side. So left side, right side. And what we're seeing here is the result of a right middle cerebral artery blood clot and its effect on the brain. So all of these areas of white are damaged areas due to the blockage of the blood vessel. So this is showing blood that's sort of like pulling, if you will, or damaging, areas of the brain that's being damaged because it's not receiving oxygen. However, in this particular case, you can see that they ended up giving this particular patient a antithrombolytic. So basically, it's a clot busting drug, and because of that, the blood function was restored, and you can see that afterwards, it's the result is actually pretty good, considering the fact. And so that's why they say in a stroke, like time is tissue, and kind of the same with a heart attack, because the faster that you can get a person clot busting drugs, the less likely, I guess, the outcomes are going to be poor, I guess, if you will. Hemiplegia refers to something that occurs during a stroke where one side of the body is paralyzed. So let's take this person, okay? and they're having a great day. They're doing their own thing. And then all of a sudden, okay, so we know that this is their left side, here's their right side. And then all of a sudden, maybe you see this side starts to droop, okay? So you ask them to smile, and this side won't smile. All right, and maybe their eyes kind of drooping, who knows, okay? But the point of this is that if this side of their body is not working, usually that means the clot is on the opposite side of the body compared to the affected side. So if the symptoms are showing up on the right side of the body, the clot is probably on the left hemisphere of the brain, because remember, we have reciprocity in the brain where the opposite side of the brain is controlling the opposite side of the body, right? So left brain is controlling right body, and right brain is controlling left body. Okay. Lastly are these TIAs or mini strokes. Basically, these are red flags for a potential stroke in the future, but they usually resolve on their own pretty quickly. So next I'll talk about Alzheimer's disease. This is a degenerative disease of the brain, and in this particular disease, eventually the patients here will develop dementia, and Alzheimer's occurs in between 5 and 15% of people over the age of 65. And so some of the symptoms here associated are memory loss, short attention span, disorientation, language loss, over time, irritability, mood swings, confusion, hallucinations are common. Essentially what's happening physiologically is that we begin to form plaques on the brain, which are basically these protein called prions. They're these little tangles of proteins, these neurofibrillary tangles. Specifically, they're called prions, which are misfolded proteins. And those proteins become all tangled up and form these plaques. And so as this happens, the brain cells die, and overall the brain gets smaller, which is what you can see over here in this Alzheimer's patient's functional MRI scan. And the brain just becomes very vulnerable. So areas that are very vulnerable to this are things like the hippocampus, the basal forebrain, and the association areas. So those areas that are highly associated with thinking and with memory. So we're almost done, so just bear with me here. Parkinson's disease is next on the list, and typically Parkinson's just presents during the 50s and 60-year-old age range. It is caused by a degeneration of the neurons that release dopamine. And so some of the symptoms here, I guess, of Parkinson's are tremors, specifically in the fingers and the wrist. Also having a forward bent walking posture called a stooped posture. Sorry about that. Oh my gosh. Here we go. So this stooped posture. In addition, a lot of times the Parkinson's patients, you'll really be able to tell when someone has Parkinson's because they have this particular gait, or gait just refers to the way that they walk. It's like this shuffling, short-stepped gait, and you can tell by their posture and their gait a lot of times when someone has Parkinson's disease. Additionally, it takes them very extended periods of time to initiate and execute a movement. And then in terms of Parkinson's, here's a big part of it, is that we don't know why it happens. So it's very unfortunate, but I know there's a lot of clinical trials going on looking for a cure, and there's a lot of research about Parkinson's looking for a cure. So one of the last things we're going to talk about here is Huntington's disease. And Huntington's disease is a fatal hereditary disorder, so it means it's inherited, that occurs during middle age, usually by 30 years old, give or take. You are diagnosed, well, now that we have the medical capabilities to test for this gene, it's usually by like 30 years old, you're able to know whether or not you have the mutation for Huntington's disease. And Huntington's disease, basically what happens is that you have these proteins that are going to accumulate in the brain tissues, in the brain cells, and the tissues themselves are dying. And so this is going to present with the initial symptoms being these really wild and jerky movements. And from the onset of the very first symptoms, this is usually fatal within 15 years of the first symptoms. The case of Huntington's is that it's actually sort of the opposite mechanism compared with Parkinson's, because in Huntington's, the motor function is actually overstimulated. And so basically, in order to sort of manage some of these symptoms, you can use drugs that are used to block the dopamine effects. But in terms of Huntington's disease itself, it's associated with particular regions, specifically the basal ganglia. So the basal ganglia. So this picture on the left is normal basal ganglia, and this is the basal ganglia of someone that has Huntington's disease. So in Huntington's disease, you get the enlargement of these ventricles as well, as the tissues of the brain are actually dying. So there you have it. Our central nervous system, specifically the brain, part 1 lecture is done. We made it. I know that was a very long lecture, and usually, like I've told most of you guys, I usually have 2 weeks to do this lecture, but because of the way that the schedule was rearranged, I had to deliver it all in 1 week, and so I do apologize, but I know that you guys will crush the test, even though this is a lot of information, and all this stuff will come up again full circle as we enter AP2, and again as we talk about the peripheral and autonomic nervous system. So don't freak out if you don't have this stuff down just yet. We will get you there, I promise. So I hope you guys have a really good day, and I will talk to you guys soon.